Hello everyone, today we will talk about bacteria and virus. Bacteria and virus have many differences. Today we will know about their history, structure, size, shape, and reproduction. So let's start and talk about history. So basically bacteria is a living thing. It is unicellular, that means it is a singular cell, and it is a prokaryote. While on the other hand, virus is not even a living thing. It doesn't have cells. In the past, organisms were divided into 5 kingdoms: Monera, fungi, protistans, animalia, and plantae. Fungi, protistans, animalia, and plantae were eukaryotes, while Monera was a prokaryote. Monera was divided into 2 organisms. They were bacteria and archaea. But later on, scientists discovered that bacteria and archaea have many differences and they can't be in one kingdom. So, they divided them into 3 domains: bacteria and archaea and eukaryotes, which include the protistans, animalia, plantae, and fungi. On the other hand, viruses doesn't have a specific history. So scientists made a theory that some DNAs came from parts of cells and made a virus. Now let's talk about the structure, size, shape, which are the important things in them. In bacteria, its structure have cell wall, flagella. Flagella is used for its movement, capsule for protection, pili, plasmid, ribosome, cytoplasm, and cell membrane. While on the other hand, virus is made only of protein coat, which is called a capsid, and DNA or RNA. For their size, bacteria is 0.1 micrometer long and 0.5-0.7 micrometer wide, while virus have size 5 to 300 nanometer. For their shape, bacteria have 3 shapes: cocci, bacilli, spirali. For virus, it have different shapes for every virus. Last but not least, we will talk about the production. The production of bacteria have 2 types: binary fission and conjugation. Binary fission is the division of a cell into 2 genetically identical cells. This division can occur quickly, as often as every 20 minutes. That means that one bacterium could become 1 billion bacteria through binary fission in just 10 hours. The other form of reproduction called conjugation, in which 2 prokaryotes attach to each other and exchange genetic information. It depends on the pilus or pili for the attachment of the 2 cells so that there can be transfer of genetic material from one cell to the other. In this way, new gene combinations are created and diversity of prokaryote population is increased. So how does bacteria infect host cell? It is by phagocytosis. First, the bacteria attach to receptors and then give orders for the host cell to engulf the bacteria itself. When it enters the cell, it is called the phagosome, and then it is part of the cell. Also, viruses have 2 types of replication: lytic cycle and lysogenic cycle. In the lytic cycle, first virus attach to receptors in the host cell, and then it injects genetic materials to host cell, either only the genetic material or all the virus. And then it starts to replicate. It gives orders to the cell and control it to make DNA and protein for itself. After the replication, we have a new virus. Then a new virus leave the host cell and start to attach to another cells. In the lysogenic cycle, the viral DNA inserts into a chromosome in a host cell. Once inserted, The infected cell will have the viral genes permanently. The viral genes might remain dormant for months or years. Then, at some future time, the viral gene might be activated by many different factors. Activation results in the lytic cycle. The viral genes instruct the host cell to manufacture more viruses. The new viruses will leave the cell by exocytosis, or by causing the cell to lyse.
The most common diseases made by bacteria is flesh-eating disease, staph infection, meningitis, pneumonia, tuberculosis, cholera, dysentery. While in virus, it is common cold, influenza, herpes, chickenpox, mumps, human papillomavirus, and measles. And that's it for today. Thanks for watching.